So now we move on to part 5. We moved on from insulin sensing to another molecule that everyone knows about, but no one really knows the structure of, unless you learn it for biochemistry, adrenaline. You might have a copy of adrenaline. I should have brought my mug. I think I have it on one of my mugs. A molecule that sort of looks like adrenaline, but is not. Caffeine is related to adrenaline. It's about the same size. And it's one of those things that people put on mugs. I guess you could put adrenaline on your EpiPen or something like that. But it would make it cost more, and Lord knows we don't need any more of that. So you can put a sticker on it. Okay. But adrenaline is a simple molecule that sends an important signal. But it works through a slightly different version. It still involves GTP, but it involves what's called a G protein coupled receptor, GPCRs. This is another of the things that if you look at job listings, there are going to be a lot of companies working on this. GPCRs are another example of something that is very different on the outside, but very much the same when you get inside. And in fact, even the outside is a little bit the same because it doesn't have big domains that bind things. These are things that bind small drug-like molecules. And if you're wanting to help people by pharmaceuticals, you're starting to think, oh, that's something that my friend Dr. Bartlett can synthesize for me a lot easier than a peptide hormone. Small binding sites for small molecules, and they all fit within these 7 helix bundles. This is already starting to sound familiar. So you have this is the way the adrenaline receptor looks. You have a ligand binding site, and then you have the receptor itself, beta-2 adrenergic receptor. Adrenergic means adrenaline making stuff happen. Ergic means making stuff happen. And then you have the different colors here for 3 proteins that all are labeled with G. And guess what? GTP is involved with this. Now, if you look at the opioid receptor, morphine analog binding to it, it looks like the same thing, different color. Small molecule, very different results, different binding site. Everything else looks the same. Here's histamine. Doxepin, anybody know what that does? I'm not a doctor enough. Sounds important. But I know histamine is important. It's important to the immune system inflammation. All 3 of these very different receptors, you overlap them, and they look like they're the same secondary structure. You couldn't tell them apart from secondary structure. They're all the same architecture outside, and they all bind the same type of protein inside. Now, they might have different interfaces to bind different types of G proteins, but they will all bind proteins that at this level look like this. This looks like a really big example of modularity that the body used to respond to small signals. Therefore, if we are looking to modify through chemistry, let's look at this in more detail. By the way, one student once talked about the beta-adrenergic receptor, and she said, before I start, I'm going to just get something set up here. And then she, when nobody was looking, she suddenly popped the balloon, and there was this big sound, like everybody jumped. It was a more innocent time. And she said that was adrenaline. So I'll just describe that to you and let you get a little adrenaline boost from just imagining it, okay? So the first and simplest example of GPCRs is the adrenaline receptor, and it's also a thing that everyone knows about. Everyone knows about adrenaline, but what receptor does it bind? Adrenaline's official name is epinephrine. That's the name of the molecule, at least. I believe adrenaline might be a little more complex, but the center of the mixture that is adrenaline is epinephrine. And that's why you have an EpiPen, epinephrine. And it looks like this. It looks like an amino acid. In fact, it's made from the same processes that make amino acids. Easy for the body to make. Distinct structure, soluble. It'll go through the blood, go to where it's needed, and cause the fight or flight response. Biochemically, what it causes is it causes you to burn stuff. It causes you to increase the breakdown of glycogen so that you have more glucose, and to increase the breakdown of fat. Basically, it gets you ready to run. That's what adrenaline does. And that's one of the reasons why, you can be shaky. And I guess it gets you ready to study as well if you're well, it's not the same as caffeine, though. So we realize that we have small molecules that have stimulant effects, but they have different overall effects because they are different molecules working at different places. Here's adrenaline in its receptor, and it binds here. It makes all these really cool hydrogen bonds. You can mutate these, mess with the receptor. You can see how that works. You can see how it fits its shape into the pocket. It's pretty straightforward ligand binding site approach. It's all chapter 5, chapter 6 type stuff. But the thing about these receptors is they do have multiple conformations. So it's not just yes-no binding. You also have, well, that's kind of weird binding. Okay? This is the weird one. This is a different GPCR, but again, they are usually the same kind of things. And there's the ligand here is pink. You can tell that it's a different shape of ligand, but the whole thing is the pink shape right here. This is the normal binding conformation. This activates the receptor. In fact, this is the ligand right here. This is kind of cool. Actually, this is the green antagonist, I should say. This is a small molecule, does not bind in the binding site. Not competitive inhibition. It's more like our uncompetitive or non-competitive, or honestly, I can't remember which one exactly because they all sound the same. But it's one of the non-competitive ones, okay? It's not competitive. Don't tell the students next year that I can't remember until I look at it, okay? So, get myself in trouble here. But the main thing is not the binding in the binding site. It's binding below it, changing the shape of the binding site, and you can see, well, that doesn't fit anymore. This is antagonism. If you have enough of the pink signal, it will still work, but it's not going to work as well. It'll have different binding characteristics, different kinetics, and a different effect on the cell. So this is the cartoon way of looking at these receptors. You have the 7 helices, you have the binding site, and you have the interface with the green G protein. And by the way, on all these slides, Leninger has kept the colors the same. I really appreciate this about Leninger, because you can follow, and the red thing is always going to be the receptor. The green thing is always going to be the G protein. So what happens is when epinephrine binds, this G protein is sitting around, it has an old GDP in it, and then it kicks that out, conformational change, kicks out the GDP, binds a fresh new GTP, and that causes a conformational change where the interface breaks. And it binds and forms a new interface to the blue protein, adenylyl cyclase. This happens to be a membrane protein. You see that the G protein here is a peripheral membrane protein. So all those parts of those chapters were setting you up for this. The other thing to notice is that the G protein actually has 3 different subunits, and only the alpha subunit binds the GTP and scoots along the membrane to bind the adenylyl cyclase here. If you think about the name adenylyl cyclase, you can start to think, oh, maybe ATP is involved, and maybe it makes cyclic AMP. That's what we have right here. That's what it does. It takes ATP and makes cyclic AMP, which is a second messenger that floods the cell. Then this goes, and it binds protein kinase A. By the way, we had protein kinase B on the previous slide, insulin. Protein kinase A, very different, but same kind of central role to signaling. Different signals, same looking network. And then it does a bunch of stuff. This is the way Leninger says a bunch of stuff happens, and even I am not going to worry you by making you even look at it. Phosphates fly around, the cell gets activated, and glycogen and fat metabolism are increased. The other part is part 7. There is actually a cyclic nucleotide phosphodiesterase. Probably pretty slow, definitely a lot slower than the time it takes for the cAMP to diffuse through the cell. But there is this sort of cAMP vacuum that's going around, and it's sucking up all the cAMP and turning it into 5-AMP at a certain slow rate. This means the second messenger is destroyed after a certain amount of time. And so the adrenaline wears off, because it's the same kind of thing as acetylcholine, actually. It's the same kind of desensitization type step. As the cAMP is degraded, it just becomes 5'-AMP, and that doesn't send the same signal. It's not the same shape, doesn't do the same thing. And so the cyclic nucleotide phosphodiesterase is sort of an eraser. So now I want to show you a video of another GPCR, but it doesn't matter because they all look like this at this level. Here's a really cool animation that I'll show you. Okay, so you have the ligand coming in, and there is, here the alpha stays, and the beta, they might have switched their terminology, so don't hang your hat too much on that. But then the GPCR goes, the G protein goes and it binds other stuff. It makes a big complex in this state, and it makes the map kinase. They tack on the Ks to indicate that there are other levels of kinases that it will affect. Look at all these conformational changes. I mean, you have all this kinase activity going on, and there you have stuff getting phosphorylated. Here they make the phosphates purple, but you know, they're just doing all sorts of things weird in this one. But then you have the kinases will go and they'll bind other kinases, and phosphates are being added that's spreading throughout the cell, and you need multiple proteins to activate later proteins. So this, by the way, is Janet Iwasa. I think I saw somebody using her book for Cell Bio. The same name. Anyways, Iwaka or Iwasa, I believe. I saw somebody using that name on a book. So let me know if there is an overlap there. But Mirko. Are you talking about the green in that video or the green previous? Mm-hmm. Right here. So the colors here are a little bit different, but the colors that Atkins keeps the same are going to be the colors for this figure right here.
What was the one before?
This one right here? Yeah, this is from a paper, so this has a different color scheme. The green here is actually an antagonist, which I would not have chosen because green goes with phosphate and green means going, but they chose it. Pink means go, and green means stop in this case.
So it's not G
protein? Well, this is actually a G. This is a receptor, so it's not the G protein. The G protein is below the receptor, right here. Yeah. So when I use something from a thing, but all the figures I use from Leninger will follow this, and that's why I want to show you right here. Here we have the green protein binding the blue protein. Remember, we have red protein, green protein binding the blue protein right here. This is the adenylyl cyclase, which makes the cAMP. So if you look at this, you can see the GTP, which is in red. Up there, the ATP is in yellow. And so GS alpha is actually a GTPase. This is the active conformation, which will bind the AC, but it will eventually chop itself off and fall off and go back to its home. Adenylyl cyclase, on the other hand, undergoes this reaction, which hopefully you have in your head already. But it will take the phosphate and it will join it to this nucleophile, and it will form the cyclic form of the phosphate. Very unique structure, very good second messenger, very soluble, easy to make. So we have that thing about cAMP binds pKa, protein kinase A, and pKa is so important, I have to show you a figure of that. Now, it turns out that pKa is very sensitive to cAMP, and it has multiple binding sites. When you see multiple binding sites, what do you start to think? Allosterically active. I think, do I have it? Oh, I have the word right here. You could just look at it. What's the big biochemistry word that we always spend way too many test questions talking about? Well, there's inhibition. Keep going. Cooperativity. Inhibition happens in lots of sciences. Cooperativity, I think that's a biochemistry thing. I'm going to claim it. Okay? But multiple binding sites cause cooperativity. In this case, it's inhibited. It's actually auto-inhibited, and then the cAMP helps it come out of its shell. You know, it displaces the autoinhibitory domain. But the main thing, it takes 4 cAMPs to bind in here and to kick off the inhibitor sequence. And then that opens up the binding site. Because 4 cAMPs are required for activation, it's going to look like the hemoglobin plot. And what is the one thing about the hemoglobin plot that you would care? What's different about hemoglobin than myoglobin as far as its binding plot goes? Brian? Yes. Yeah, and what's the difference in binding slope when you have that? You have the sigmoidal characteristic, was what it looks like, Mirko? Mm-hmm. Yeah. Right. Right. So you can have different kinds of. Now, negative cooperativity is where you make the slope less steep. But usually this is positive cooperativity, where you are increasing activity the more stuff you have. And that means the slope is increased, which means as I increase in CAMP concentration, I'm not going to have like halfway producing signal. It's either going to be on or it's going to be off, because it's such a steep curve. You're moving along the graph, 0, 0, 0, 0, 0, 100, and then you're all the way up there. That's what cooperativity gets to you. And that's why CAMP has 4 binding sites here, because you get a more sensitivity to CAMP concentration and a more defined result. You get either 0 or 100. You don't have a wishy-washy slope where it's like 40%, 60%, 80%. Okay? So this is the actual structure of this. And by the way, this keeps the same pictures of this. See how you have, this is the catalytic domain that will actually do the kinase thing. This is the AMP binding domain. That's in red. And you see right here, this is a big inhibition of the cleft. And so it's really blocking the substrate binding cleft. AMP binds here. This falls off. And then that can go around and activate all the other proteins in the rest of the cell. Zach? Well, let's see, we're talking about, well, this is adrenaline, but it applies to most of these. Most of these, you don't want the cell in an in-between state. You want to be either sending the signal or not. You want to be either sending the adrenaline signal and doing it. You don't want it accidentally halfway turned on, for one thing. And you want to be definitely sending the signal, definitely sending CAMPs. And I think it's probably mostly a safeguard against accidental activation more than anything else. So this is another look of how CAMP actually binds. This is the actual binding site. Again, it's from another paper, so the colors are all screwed up. You can imagine this one being red and this one being blue. This would be the catalytic subunit. It's definitely blocking it right here, right? And then this opens it up to bind other proteins. So you can actually look through this pathway, and you can look at the stuff after pKa, and we can see that actually this is the stuff after pKa in the glycogen phosphorylase. So this is after pKa, it's going to break down glycogen and produce blood glucose. How much glucose can we produce from one epinephrine molecule? Well, let's just say that we can produce 10 GS alpha molecules, which by the way we can. Once this one has left, another one can come in, and you can actually produce 10 molecules. Each of those, conservative estimate, can produce, can turn on an adenylyl cyclase, which can itself make 20 molecules of CAMP. And so we're already to 200 molecules from one. Then we go to pKa, and because of the cooperativity, that actually turns down the signal a little bit. Again, this is not about always amplifying the signal at all costs. It actually, it's a more sensitive signal, but it's also just like with cooperativity, actually hemoglobin binding, oxygen binding, is lessened for having a steeper curve. And the same thing is going on here. You have overall binding, percentage binding, lessened, but you have a steeper curve, which is not reflected in this graph. All you see is the lessening, where it ends up with 100 molecules. But then you have an active protein kinase, and again, let's be conservative. Let's say that it can only activate 10 other kinases. Each kinase, 10 other kinases. Well, I have 100 of these, so then I'll have 1,000 of the next level, and then that will actually be able, each of those activates 10 glycogen phosphorylases, which is 10,000 of those. And even if each glycogen phosphorylase can only liberate 10 glucose molecules, we still end up with 100,000 molecules from this signaling pathway. And if those all go into the glucose, you have now produced 10,000 molecules from one molecule signal. That's a pretty good deal, you know. So, and the other thing is, this is just pKa doing one thing. pKa has a lot of other pathways to activate as well. You could probably do a similar diagram for its fat breakdown result. In fact, you can look at what pKa binds, and it binds all these things. These are sequences that will be directly phosphorylated by pKa. And so you see glycogen synthase, and you can see all of them have this serine that it really cares about. But everything else is slightly different, but these all bind the same site. These all will be activated by pKa. So you can look up and down that list and say, you know, what is most surprising to me, what is like my research, you know, things like that. It binds histones, for goodness sakes. It binds pyruvate kinase. You can look up and down, and you can see all these different things that it activates. So just remember that pKa is turned on by cAMP, but we also have this desensitization procedure where you have a cyclic nucleotide phosphodiesterase. It's a mouthful, but it's specific. If you can translate it, you can see what it does if you know the organic chemistry without even looking at the structure. You can know it takes a cyclic nucleotide and it breaks a phosphodiester bond. So it's going to do this to it. And that will turn off the signal. It's the eraser. Notice right here, caffeine shows up again. Caffeine actually does something inside the cell. Because it's inside the cell, it's probably not as important, but it's definitely something, you know, so it's a biological question as to which one is more important. So it's how this pathway can be turned off. So realize that we've already talked about several ways to turn off this pathway. GS-alpha has a GTPase activity. The cyclic nucleotide phosphodiesterase has its own activity. Those are two ways to turn it off. There's another protein that goes around and it looks for beta-adrenergic receptors without the GS-alpha, and it grabs them and it pulls them into the cell. It's called arrestin, pretty good name, and it's internalized, and you can actually see that in a figure in Leninger. And then remember there's a bunch of G protein coupled receptors. By the way, this is a typo I always do. I just have a mental block. This is wrong. It's G protein coupled receptors, but I get the coupled and the protein messed up. Anyways, that's all right. You can still explain yourself clearly even if you do mistakes, okay? But some of them bind other receptors that are bound to G proteins, but these are G proteins that will go and bind to adenylyl cyclase, and then they'll just sit there and block it. So we don't call those GS-alphas. They aren't activating it. They're binding in the same place. They have the same architecture, but they aren't proceeding. They're inhibiting. And so these are called GIs, and I think of them as the evil twins to the GS-alphas, okay? GS will turn the second messenger on, but there can be other things, good example of integration, GIs that go in and turn it off. Which brings us to the end of how these pathways work. Now we are going to really bring it out. So give me a second to switch.